Now we will discuss starch synthesis. As we have discussed that the starch is very predominantly important quality criteria for many products, especially for tubers as well, especially in potatoes as we earlier said. So let's see what are the different processes that are involved for this synthesis of starch. So generally this happens in plastid. Starch is all synthesized in plastids or amyloplasts when the tubers are initiated during the very early stage of growth. And then these tubers start growing, and the time when they start growing, the number of these starch would actually increase. The particle size of the starch would also increase. So there would be more number of starch grain, there would be bigger starch grains as well. So this is the growth process in all the tubers. As we earlier looked at, that these starches all consist of two types of subunits. Those are amylose and amylopectin. So the amylopectin is highly branched. The amylose content are relatively unbranched component of them, and when they join together they make up different kind of starches. And if you look at the relative proportion of these, it is 21 to 25% amylose, and then 75 to 79% amylopectin. This is present in most of the starch. Particularly this is very true in most of the tubers. Again, it would depend upon different kind of cultivars, this relative proportion, and we have to actually try to modify them according to our own requirement through biotechnology as well. So starting this process, it would start with this ADP glucose. This would be synthesized into starch by the different action of enzymes, because glucose is the basic unit. This would start joining together and through polymerization, and there are different enzymes which are responsible for these reactions at the very beginning. So one of the important one is ADP glucose pyrophosphorylase, or ADP GPase. This is the enzyme which is important. And the second enzyme is starch synthase. This has got very important role to play for the formation of starch in different tubers. And then there are starch branching enzymes as well. So we'll see some of the detail about these enzymes, that how they convert these glucose into starches. So it would start with the conversion of sucrose into hexoses, hexose phosphate in the cytosol. So sucrose would be broken down into hexoses. Then we have glucose 6-phosphate. This would be transported into amyloplast, where it is converted into glucose 1-phosphate. So this reaction would take place in the amyloplast, and then this glucose 1-phosphate, it would be converted to ADP glucose by the action of ADPG GPase enzyme. So this is the pathway for the formation of most of the starches. And then the different other enzymes, as we said, starch synthases, they have got their own role. They would be catalyzing the polymerization of glucose monomer into alpha-1,4 glucons, and this would use ADP glucose as a substrate. So basically this is joining these glucose molecules and 1,4 linkage. As we in the initial lectures look at the chemistry of these starches, that how these glucose units are linked together, this 1,4 linkage. And then the starch branching enzymes, they start playing their role, and they would be making this linkage at 1,6 linkage level. As we earlier looked at, that most of the branching that happens in these chains of glucose molecules, this is at the branching level, this is 1,6 linkage. This is developed in the case of amylopectin. As we know that amylopectin are more branched polymers, so they would have more branches, and these branches are they utilize this 1,6 linkages between two glucose molecules. So if you look at this all reaction, because we know there are different enzymes involved, but this ADP GPase enzyme This is generally considered as the limiting factor for production of starches, and it is due to this allosteric activation by 3p glycerate and inhibition of inorganic phosphate. So this would play its role in these two modifications, and due to which this would be very much rate limiting effect of ADGPase enzymes. So if we can control, or if we can know the exact mechanism of these enzymes, or if we can activate, or if we can express these the genes that are responsible for this kind of enzyme, we can modify the route and the mechanism for formation of tubers, starch in tubers. So as said, we need to look at the strategies, like how can we alter this starch metabolism by manipulation of this one particular enzyme, and that would be again deciding that what is the level of this reducing sugar that is produced, whether desirable or undesirable, and how we can tame this natural mechanism to our maximum benefit, that they can be stored properly, or they are not having they are not very much affected by different storage conditions, etc. So this is the one of the approach that biotechnologists can utilize to look at this particular enzyme that has got a very strong role to play in the end quality of the product, especially in tubers for synthesis of starch.